Hey guys, welcome back. Today we're going to be looking at the second part of our recombinant DNA series. So in the last video, we really looked at sort of the anatomy of these different types of expression and shuttle vectors. So today we're going to be using them to do a couple more advanced things. Okay, we're going to be able to relate this to some of the genomics techniques that we learned in earlier chapters. So the main focus today is going to be on the creation and use of what's called a library. So I'm sure you guys know what a library is in a general sense. Sometimes thinking about a library in a genetic sense can be a little bit confusing, but really they're the same thing. Just like a library is a collection of a lot of different books and resources, a genetic library will be a collection of all of the DNA in an organism. So this can be genomic DNA, which we'll talk about, or this can be cDNA, which again is going to be representative of all of the gene expression that's happening in different cell types. So today we're going to be looking at kind of how to create them, what are these libraries, and then we're going to use them to screen for cDNA protein expression. We're going to be looking at finding genes, and then we're going to be using them to validate gene function. So we've got making them, and then 3 good uses for these guys. So let's go ahead and get started with how to create a library. What is a library really? So if we're going to start out with a DNA library, we need to use the DNA from the organism that we're trying to study. So we'll start out with a genomic DNA library because that's the most straightforward, and we'll look at how that goes. So we're going to need the whole genome from whatever organism we're working with. Okay, that way it will contain all of the genes that we'd be interested in seeing. Okay, so we're going to be kind of isolating out different parts of this genome. So first we have to break it into smaller pieces. Okay, so we'll break these smaller pieces, and these can usually maybe be several thousand nucleotides long, so there are really a lot of pieces here. And then we can put the pieces into the vectors, kind of like we looked at last time. Okay, so these would be more like shuttle vectors, taking these pieces and dropping them down into some type of vector. Okay, so maybe a shuttle vector, or we can plan ahead when we get ready to do some cDNA work, making sure that we can express proteins from them. Okay, but for now, hopefully what you can observe is that in each of these vectors, we have a slightly different color piece of DNA that should correspond to different pieces that we have kind of over here. Okay, you can see that we've broken up these pieces and then ligated or glued them into our vector. Okay, so in principle, there would hopefully be representation of every gene that's potentially found in this organism. Okay, so that would be quite a lot of these vectors. Okay, and this is controlled using, generally speaking, kind of a stoichiometry approach. Okay, so we've got hopefully a vector that's got a piece of a gene. We're going to represent the entire genome. Okay, and so again, each chunk should be unique. You may have some overlap, but we should at least have some type of unique representation here. Okay, so now we can put them to use. So a general way that we can use these is to take these plasmids, these vectors, and put them into bacteria. This doesn't always have to be bacteria, but this is a pretty good starting host. Okay, so we can put them in a bacteria, and we'll be working with that a little bit later. Okay, we can also have variations on this theme that will include the type of vector that we're going to be using. So we'll see not always bacteria, and sometimes again we will have cDNA instead of genomic DNA. So that would again represent all of the genes turned on in this genome, okay, instead of genomic fragments. So they would be ready to produce protein. Okay, so we've got a couple of things we can do. All right, so let's go back to that bacteria example. So let's say, okay, our vectors have gone into the bacteria, and you can see that again, each of these bacterial cells contains a different vector, okay, a different piece of this genetic DNA. Okay, this genomic DNA They're not doing anything with it necessarily right now, but we'll get to that part later. Okay, so these vectors in these bacteria will be plated out on selective media to ensure that they contain one of these plasmids, these vectors. And once they're there, we let them just hang out, eat, float, and replicate, which is what bacteria do. Okay, so we're not using this library yet. We are still creating it. Okay, and so if you notice, we have populations that contain identical things kind of all together. This is what we call colonies. So if you've taken microbiology or you've worked with bacteria before, you may know what the colonies are going to look like. Okay, so these are going to have all of the identical fragments hanging out in here. Okay, so we will take these colonies and store them in individual wells of a plate. Okay, so all of those colonies from the last slide we can see are stored individually. They all contain the exact same thing within this colony. Usually when you create these types of libraries, these can be hundreds of wells, sometimes thousands of wells. So there's really a lot going on here. Okay, we don't have so many, but we'll deal with these that we have and extrapolate out. So now that we've created this library, these are in bacteria. You can add some solid glycerol and store them in the -80 until you're ready to use them. Once you're ready, we can do a couple of things. Okay, so the first example that we're going to look at is going to be screening cDNA libraries. Okay, so the important part about this is that the type of vector that we'll have to choose needs to be set up for expression. We will be taking cDNA from whatever organism that we're interested in. Okay, so this is representative of expressed genes, and we're putting it into some type of expression vector. Okay, so the bacteria, in this case, will have been hard at work making these proteins for us. Okay, when we start to plate these out, they'll be making these proteins. Okay, so we can actually print copies of these bacterial colonies in the exact orientation that we see here onto new plates. That preserves your original library. This is like basically making a copy of something if you go to the real library. You want to leave whatever reference material it is in the library, but you want to take a copy to go, and you can write on your copy, you can highlight on it, do whatever you need to, but you're leaving the original alone. That's the purpose that we see here. Okay, if we know then exactly the protein that we're looking for, which this does require some planning ahead, we will hopefully have some antibodies for this. Okay, so we will take these antibodies, which again will have an affinity for a very particular protein, and these will be radioactively labeled. So we just dump some radioactively labeled antibodies for our particular protein onto these membranes that we have. Okay, we'll lyse the cells so they'll leave behind their protein, none of the other junk, and then dump these antibodies there. Okay, so hopefully one of these antibodies will recognize the protein we're looking for. Okay, so the key here is going to be that our antibody is going to need to stick to the proteins that we see, and so only the cell that actually is actively expressing our protein should be visible. Okay, so maybe it would look something like this, and so we can say, okay, the radioactive antibodies are bound right here. That means that our protein was here. We can go back to our original plate, say, okay, this was the guy. And so the point of this would be to pull this particular colony out so that you could study it further based off of the proteins that it was making. Okay, so in our next example, we can make a similar kind of assay by, instead of going for the protein, we can look for the genomic sequence instead. So screening libraries, we can look at a genomic library. So let's go back to that idea of taking our DNA, our genomic DNA, breaking it up, and we clone these in sometimes into vectors But a good variation on this, just for example, is that we can also put these little pieces, trap them into these little phages. So each of these phages, hopefully you can see, gets a different piece of DNA. So just because this is another example of a way to do this, another example of an approach, we'll go ahead and continue on with our phages. So phages we can store on plates in little plaques, okay? And so each one of them, they're not going to be expressing proteins. That's sort of the virus phage lifestyle. They're just going to be containers for this DNA. So we can keep them, again, as plaques, and on this case, grow them up and then put them onto a membrane, just like before, where each one is kind of represented here, remembering that what we care about is the DNA. So once this is on a membrane and we're ready to screen the library, we can wash these proteins away, leaving their DNA, and then we can start to detect it. So in our protein example, what we used was going to be antibodies. So we're going to use antibodies, but in this case, what we're going to be doing instead is using complementary DNA sequences called probes. So again, in principle, we would know exactly what we're looking for here. So we would produce a DNA probe that is complementary to only that sequence, and we would radioactively label it. So this is going to be our radioactive probe. Remember that it's only going to be complementary to one of the sequences here. So once we've got all of our probe on here, we can let it incubate and hybridize, and then we can wash the excess probe away. Only the bound probe will stay, and it will let us know where exactly that fragment is. So this would show us that the probe was complementary to the sequence. So this is going to be where we need to look. We'll have to then go back to our original membrane plate, restoring these guys, and say, okay, this is the one we wanted, so now we can work with it. Maybe we want to sequence it further. Maybe we just need to do something with it, but now we found it, okay? So the last use of libraries that we're going to talk about in this video is the complementation assay. So we can do this using libraries. This is one good way to use them, but complementation, just in a general sense, is going to be something that scientists use a lot and still continue to use, even when we know what the gene is, using some of these recombinant DNA technologies that we've been talking about previously. Okay, so in this particular context, the time that we'd want to do a complementation assay would be when we want to find out what gene is responsible for a particular phenotype. So let's say, for example, we have a large population of yeast, Saccharomyces cerevisiae. Okay, this is also brewer's yeast. Okay, and so maybe a phenotype that we might find is one of these guys can't make arginine. Okay, so this is kind of like those autotroph mutants that we talked about in the beetle and tatum experiments. Okay, so one of these guys can't make arginine. The background behind this is we don't know why. We just know that it can't make arginine. So we're about to find out what's going on. So again, we would transform our genomic library or our cDNA library. Okay, probably cDNA in this case because we want to get expression from these. But again, representing the entire collection, if possible, of expressed genes in yeast. Okay, so each one gets a different gene. Okay, these are going to be living yeast. They're going to have their own chromosomes, but we're going to drop our vector in on top of it. Okay, knowing that these yeast, every single one of these guys on this slide, cannot make arginine, what we're going to look for is, once we get these plated on media that doesn't contain arginine, we're going to look for the one that actually can live. Okay, so what that means, all of these other guys, they can't make arginine. They couldn't when we put them on this plate. we did not give them any arginine, so they died. But one of these guys, curiously, is still alive. And what that means is that whatever piece of DNA that we've put here, we can now express a gene that will allow us to make arginine, ARG1 for arginine synthesis, likely the culprit. But now we know we can take this out, we can sequence it, and now we're like, okay, this is the reason they couldn't make arginine. And now we know a little bit more about the arginine biosynthesis pathway. We can isolate this and study it further. Okay, so some conclusions to take away from this. Libraries can be made to store an organism's cDNA, so express DNA, or just a regular genomic DNA. Okay, we can screen these libraries to find out more about things that we're interested in. We can use them to learn more about what is causing a particular phenotype. And we can also use these libraries to demonstrate a gene's function via complementation. And so this general idea of storing kind of all of this functional information about a different organism, this can be expanded out into things like protein-protein interactions or transcription factor interactions with DNA. So you can make all kinds of different libraries depending on what types of interactions you're going to be interested in. Okay, so go ahead and end here with our biology joke of the day.